皆様、リンク J のライブウェビナーにご参加いただきありがとうございます。本日はリンク J UC サンディエゴジョイントウェビナーシリーズ第6回 WIS 慶応大学再生医療と計算神経科学における最新の展望セッション3をお送りいたします。私は本日司会を務めますリンク J の関と申します。本日は画面でお示ししているプログラムに沿って進めてまいります。ご質問はウェビナーの Q&A 機能からお願いいたします。時間の都合上、すべての質問へのお答えができかねる場合がございます。また、本イベントでは、ウェビナーの同時通訳機能、英語字幕をご利用いただけます。なお、ウェビナー中の録音、録画、画面キャプチャーなどはお控えください。それでは、リンク J 常務理事曽山と UC サンディエゴ国際アウトリーチディレクター和賀様よりご挨拶いたします。よろしくお願いいたします。
はい、皆様おはようございます。今日はですね、リンク J のウェビナーにご参加いただいて誠にありがとうございます。ご案内の通りですね、UC サンディエゴ校とリンク J はですね、これまで、去年の暮れぐらいからですね、UC サンディエゴ校のですね、トップ研究者の方にご登壇いただいて、かつですね、共同研究をされているその分野で、いらっしゃるですね、日本のトップのですね、研究者の方にもご参加いただいてですね、セミナーをシリーズ開催してまいりました。そして今回ですね、シリーズの第6回目ということで、UC サンディエゴ校のマーク・トゥジェンスキー先生、そして慶應義塾大学の牛場純一先生、このお二人の先生がですね、脊髄損傷に対する、幹細胞治療や脳卒中後のリハビリ、こちらに向けてということで、ブレインマシンインターフェースの新展開、についてお話をしていただけます。両校のですね、アンドリュー、マクロー先生、そして、リンク J 理事長でもいらっしゃいます岡野秀幸先生がモデレーターをしていただきます。そしてディスカッションという中身になっておりますのでよろしくお願いいたします。それではですね、シリーズ、いつもですね、やっていただいているんですが、UC サンディエゴ校のオフィスオブアウトリー、オフィスオブリサーチアフェアーズのインターナショナルアウトリーチディレクター、和賀さんからですね、イントロダクションとモデレーターの先生方のご紹介を改めてお願いいたします。和賀さん、よろしくお願いします。
はい、おはようございます。Thank you, Soyama-san, for the warm welcome. I'm Miwako Waga, Director of International Outreach at UC San Diego. I am very grateful to Link-J Soyama-san and his colleagues who host this series of joint webinars, inviting excellent speakers from UC San Diego and our partner institutions in Japan. Keio University and UC San Diego are in the process of expanding academic research collaborations to include engineering and technology in addition to medicine. Today, Professor Okano, Dean of the Graduate School of Medicine at Keio, and Professor Makalo, Director of the Institute of Engineering in Medicine at UC San Diego, will moderate the session. Following Okano-sensei's remarks about the collaboration, Dr. Makala will briefly talk about IEM and collaboration and introduce the first speaker, Dr. Mark Tuzinski. Let's welcome Okano-sensei to the virtual podium. Okano-sensei, please take the mic.
Okay, hello everybody. Good morning for the audience in the East Coast and East Asia, and good afternoon for the audience in the West Coast, and good evening for the audience in the East Coast. Okay, so thank you very much for kind introduction. My name is Hideki Okano, the Dean of the Keio University Graduate School of Medicine. Okay, so this is part of the Link-J and UC San Diego joint webinars. So this is a joint webinar between the UC San Diego and its partner institutions. Keio University has been working with UC San Diego for these 6 years. And so this is a third series of the webinar between the Keio and the UC San Diego's. One of the very important focus is regenerative medicine and computational neurobiology. Okay, so in the couple months ago, we had webinars by inviting the Dr. Masaya Nakamura in Keio University and so Larry Goldstein in the UC San Diego. And the second session we invited Terry Shuanovsky in Salk Institute and also Yasue Mitsukura in Keio University, the Faculty of Science and Technologies. This time, we very fortunately we could invite very active and prominent scientists from both sides. We invited Professor Mark Tezinski to UC San Diego, who is the authority is the regenerative medicine in the field of the central nervous system. And so we could invite young, brilliant scientists from Keio University, Faculty of Science and Technology, Junichi Uchiba. And he's an expert of the brain-machine interface and system neuroscience. I hope that you could enjoy this webinar series. Thank you very much.
Arigatou gozaimasu. My name is Andrew McCulloch, Director of the Institute for Engineering and Medicine at UC San Diego, and it's my pleasure to introduce today's first speaker in this third joint webinar between Keio University and UC San Diego, hosted by LinkJ, on the themes, as Okana-sensei mentioned, of computational neurobiology and regenerative medicine. And I must first express my gratitude to Soyama-san at LinkJ, Waga-san at UC San Diego's Office of Research Affairs, and Okana-sensei and his colleagues at Keio for all the work that they have done to put this series together. And this third webinar promises to be just as exciting as the first two, and from all accounts is also the biggest with over 300 attendees. As Okana-sensei mentioned, our first speaker today is Professor Mark Tezinski, who's Professor of Neurosciences at UC San Diego and founding director of the Translational Neuroscience Institute here. Professor Tezinski was trained at the University of Minnesota, where he got his undergraduate degree in MD, then did his clinical training in neurology at Cornell University Medical Center, and then came to UC San Diego, where he did his PhD in neuroscience. He is a highly accomplished and well-known physician scientist whose research focuses on the central nervous system and its plasticity in animal models. But he's also translated his lab's discoveries to the clinic to help patients with Alzheimer's disease, spinal cord injury, and peripheral nerve injury. His lab uses a variety of high technology techniques, including stem cells, tools for gene delivery, and bioengineering approaches. He was the first investigator 20 years ago to start the first human clinical trial of a gene therapy to treat adult human neurodegenerative disease, and he's the recipient of numerous awards. I'm very grateful to Mark for taking the time to talk to us today, and we're looking forward to his talk on stem cell therapy for spinal cord injury and its transition to the clinic. Mark?
Hajimemashite, watashi wa Mark Tezinski desu. Thank you very much, Dr. McCulloch. Thank you, Dr. Okano. It's a great pleasure to be here and to have this opportunity to tell you about our work. So I will start my PowerPoint. I assume that everyone can see this. So I'm going to talk today about these potential stem cell therapy for spinal cord injury and our effort to translate this to human clinical trials. And I'd like to acknowledge at the outset a number of people in my group who have contributed to this work, especially Paul Liu, who really encouraged us to begin work on stem cells, Gunnar Poplavsky, who did transcriptome analyses that I'll talk about, Efren, who's been involved in monkey studies, and Hiromi Kumamaru, who developed the cell line that we'll be bringing into humans. And our work is supported by the US Veterans Administration, NIH, and a number of other foundations, and I have no conflicts of interest. So the problem that we are trying to address is human spinal cord injury. There are approximately 15,000 new injuries per year in the United States. There are 500,000 people living with chronic spinal cord injury. And this tragically often affects people early in life and results in a complete loss of function below the level of the injury. and there are no treatments to repair the injured spinal cord. So why doesn't the spinal cord regenerate? We now understand broadly that it is due to several mechanisms. Among these is the lack of a substrate in a lesion cavity to which an injured axon can attach and grow. Secondly, there is insufficient production of growth factors in the nervous system and appropriate spatial and temporal gradients to support new growth. Third, there are a number of proteins in the central nervous system associated both with myelin and the extracellular matrix that inhibit growth potently. Inflammation contributes to the degree of injury. Then finally, there are questions about whether an adult neuron, after terminal differentiation, is still capable of re-entering an active growth state after injury. So we worked on the problem of spinal cord injury for a number of years. And in 20 years of research before we began working with neural stem cells, we succeeded in regenerating approximately 100 axons into and beyond a site of complete spinal cord transection in a rat. 100 axons from an original pre-injured population of 1 million axons. So in this, I show you our best success up until we started working with neural stem cells. This is the rat spinal cord, and it is a horizontal tissue section that is 35 microns thick. The rat's head is to the left and the tail is to the right. And this is a complete transection site. And we have grafted into this site fibroblasts that have been genetically engineered to make a nervous system growth factor called BDNF. We also injected viral vectors beyond the lesion to act as a growth factor gradient to pull the axons across. And these are the injured axons coming from the brain that control motor function in black. So you see these axons regenerating in very small numbers into the graft and then some growing beyond. And the best we did was to get 100 axons to grow 1 millimeter beyond. Now we enter the era of neural stem cells. Oops, I'm trying to advance and I can't. And this is what we see today. So again, this is a horizontal spinal cord section. This is the T3 complete transection in the rat. The head is to the left, the tail is to the right. And this is a graft of human neural stem cells derived from human embryonic stem cells. The stem cells have been genetically modified to express green fluorescent protein. So we can unequivocally see the cells and the axons that they extend. And here we have tens of thousands of axons, not 100 axons, tens of thousands of axons growing not 1 millimeter, but 50 millimeters, 50. And when we place these grafts into non-human primates, we see growth of 300,000 axons for distances of 50 millimeters. So this exceeds the potency of all of our work up to that point by manyfold. This is an example of the remarkable biology of neural stem cells in the context of spinal cord injury. So I'll take you through some of the details of this now. And again, contrast this to the limited growth we saw previously. So I think many people on this call know very well what stem cells are. I think some don't, so I'm going to give a brief introduction. All of us began as a single cell, an embryonic stem cell. And a stem cell is a stem cell because it is self-renewing. It is capable of dividing and continuing to divide indefinitely. But during human development and the development of any species, some populations of the embryonic stem cells go down a pathway of differentiation and commit to a neural fate, to generating the neurons and glia of the nervous system. And these are called neural stem cells. They're still capable of self-renewing. They can undergo many rounds of division. But at some point, many of these cells commit then to become an intermediate neural progenitor, which is developing less division potential. And this is a transition state to becoming a either committed neuronal progenitor cell or a glial progenitor cell. And then these further constitute the mature neurons, astrocytes, and oligodendrocytes of the adult nervous system. So this process of development in the human takes more than a year. And the cells that we work with are generally at the stage of neural stem cells or intermediate neural progenitors. We are starting with cells that can reconstitute a neuronal and a glial environment in the injured spinal cord for reasons I will show you in a moment. This is our hypothetical model for our approach to using neural stem cells in spinal cord injury. So spinal cord injury is characterized by a cystic lesion site, and axons shown in red are transected. They are interrupted by the spinal cord injury. In our hypothetical model, we will graft neural stem cells into the injury site to reconstitute a neural milieu or an environment. And in this hypothetical model, host axons that have been cut will regenerate into this graft and form new synapses onto the grafted neurons. The grafted neurons, in turn, in the model, will extend axons beyond the injury site and to host circuits below and form synapses. So in this way, they will form neural relays. It's like cutting a wire and then splicing together the wire. This is the hypothetical model. So in our experiments, we generally start with embryonic stem cells that we drive to a neural stem cell fate that, again, are capable of generating neurons and glia. We perform spinal cord lesions, and then we implant the cells 2 weeks to a month after the injury. And we choose this delay because it's more clinically relevant for human translation. The cells also survive better if we do a delayed graft than an acute graft. Now, it took us some time to develop the technology for these grafts to actually survive. And the way we worked that out was to embed them in a fibrinogen thrombin matrix that rapidly gels when placed into the lesion site. And we include these growth factors and an anti-apoptotic molecule to support their survival. Then we graft these to various injury models in mice, rats, and monkeys. And then we examine outcomes weeks to years later. And the story I'll tell you now is that when we do this, this hypothesis holds. We see robust extension of axons from the stem cell implants in the injury site. Host axons regenerate into them and form synapses. And this results in improvements in function that is leading to our translation to a human clinical trial. So this is what we start with. This is a complete transection at the upper thoracic level of the spinal cord. There is no spared tissue. We see the lesion site. And then this is a graft of GFP expressing, in this case, neuroprogenitor cells placed in the injury site. And 6 weeks later, we can see that many of these cells mature into neurons expressing NUN, and many of the cells mature into astrocytes. If we start with human neural stem cells and look at their maturation and their fate after 6 months in the injury site, approximately 80% become neurons, about 10 to 12% become astrocytes. There are some oligodendrocytes, but the oligodendrocytes mature much later on a human time frame of neural development. If we looked at the cells that become neurons, the majority of them become excitatory neurons expressing CAM kinase. Some become alpha motor neurons, and some become inhibitory GABAergic neurons. And again, when you place this in the lesion site, you see this abundant outgrowth of axons shown at higher magnification here. You see parallel arrays of stem cell-derived axons growing into the host spinal cord below the lesion. The red is neurons indicating gray matter, and you see these axons in the gray matter, as well as what had been the inhibitory white matter. We can also start with human-induced pluripotent stem cells. So we collaborated with Larry Goldstein. He generated these iPSCs from an 87-year-old man, and we implanted this in an immunodeficient rat at the C7 level. And here, several levels below the injury site, we see 87-year-old human axons descending down the injured half of the spinal cord. Many of these axons penetrate the gray matter, and as I'll show you, form synapses. Again, this is a higher magnification view of this remarkable biology. Everything green here is an 87-year-old human axon growing down the injured spinal cord, branching off, as you can see here, and intercalating or interposing itself among the neurons that are labeled blue here in the host gray matter. These emerging green axons come close to the cell soma. They colocalize with synaptic markers, synaptophysin, and they form synapses, proven at the electron microscope level. These are asymmetric, excitatory synapses. And we see the same thing occur in the Rhesus monkey spinal cord also. So the graft survives, it extends axons down, and they form synapses. For this to be a functional relay, the host must also regenerate axons into the graft and make synaptic contacts. Do they? Yes. So this is the host corticospinal projection. This is the projection from the motor cortex to the spinal cord that is absolutely essential for human movement. It was very hard in previous work to get these axons to regenerate into an injury site. But here you see these axons are labeled red as they descend from the brain. They encounter a graft in the lesion site, and this is all of their growth into the lesion site. So now they regenerate. If you give them an appropriate target, then they regenerate. They regenerate into a spinal cord stem cell graft, but they do not regenerate into a forebrain stem cell graft. They require the proper target. They form synapses. And if we inject channel rhodopsin into the host motor cortex and then prepare a slice and excite it with light, then we see an excitatory postsynaptic potential that is generated and many action potentials. So these are synaptically active. Which host systems regenerate into the graft? All host systems that normally regenerate and that normally grow into an intact spinal cord also penetrate the graft. Do these regenerating host axons into the graft form an appropriate cell type in the graft, or do they make inappropriate connections? Yeah, I don't have time to go into this now, but in 2 published papers, we have shown that they generally find the correct type of target. Another remarkable feature of the cell biology that the injured adult axon recognizes guidance cues in a developing stem cell environment and forms appropriate synapses. So are these functional? So after a complete spinal cord transection on the 21-point BBB scale in a rat, if we implant neural progenitor cells, then we see a significant improvement in movement of the hind limbs compared to animals with no graft over several weeks. Then if we recut the spinal cord just above the grafted implant, thereby cutting host axons growing into the graft, then we entirely abolish the recovery, indicating that we have formed a true neural relay. We can also prove this electrophysiologically, but in the interest of time, I'll move on. If we do a mid-cervical injury in the neck and implant neural stem cells, then compared to lesion controls, the stem cell grafted animals also show significant recovery. It is not complete recovery. It's about recovery of 40% of overall function, but it's much better than lesion controls. And this could be meaningful for quality of life with people with spinal cord injury. So based on this and other studies, we decided to move this into a Rhesus monkey model of spinal cord injury in collaboration with our colleagues at UCSF, UCLA, UC Irvine, Davis, and Colorado State. So Rhesus monkeys are big monkeys. They weigh about 30 pounds. And I place a spinal cord lesion in the monkey spinal cord. We lesion the right half of the spinal cord at C7. This enables monkeys still to walk. They have bowel and bladder function. They are functionally fine, other than having a weak right hand. And this is our functional assay for detecting improvement. So we place the lesion. 2 weeks later, I open the spinal cord, and this is the injury site, and I implant our neural stem cells in a fibrinogen thrombin matrix into the lesion site. The monkeys are immunosuppressed with human immunosuppression drugs, and then we follow them over time. And here's a graft showing nice survival and fill of human embryonic stem cell derived neural stem cells driven to a spinal cord identity in the Rhesus monkey. And here are axons emerging from the injury site in the tens and hundreds of thousands, replicating the biology we saw in rats. And that from the C7 injury site, if we look at T1, then we see many axons continuing down in the lateral white matter. So everything green here, once again, is a human axon descending in the primate spinal cord. And these human axons are inserting themselves among the alpha motor neurons of the spinal cord that control movement. And they form, as I showed you earlier, synapses and asymmetric synapses that indicate that they are excitatory. Other host axons, or rather, I'm sorry, host axons penetrate these grafts just as in the rats also. So everything red here is a host neurofilament labeled axon penetrating to the core of this graft. And if we do a functional assessment in the monkeys, seeing if they can use a pincer motion to extract food rewards placed into these Brinkman boards. And if we look at their ability to use the right hand, then we can assess functional outcomes. So without a graft, the right hand is clubbed and the monkey cannot extend the fingers. In many monkeys that receive grafts, they are able to extend the fingers again and grasp a food reward like an orange. So on a functional assessment scale, looking at their ability to use the hand and manipulate objects, we see again about a 50 to 75% improvement in function compared to lesioned control animals. Again, it's not complete recovery. We consistently see about this 50 to 100% improvement compared to the lesion control, but it is not complete recovery. Nonetheless, it is highly significant statistically, and this could translate into somebody with a high cervical lesion being able now to move the arm and control a motorized wheelchair, or someone with a lower cervical lesion now getting enough finger movement that they can begin to manipulate objects, hold a glass, do something of that sort. So we consider this a significant enough effect to justify moving into human clinical trials. To do so in collaboration with our colleagues, Beatty Bresnahan at UCSF, they developed a monkey contusion device. We now are placing grafts into hemi-contusion sites in monkeys. So this is a graft 9 months later. Again, it's a right-sided contusion, so the monkeys can still use both legs. They can use the left arm quite well. They have normal bowel and bladder function, but they have a weak right hand. And that is our assay for looking for functional improvement. And indeed, we see axons grow down and we see and we're assessing improvement in function. So these preclinical studies are ongoing, and we are optimizing methods for graft survival and fill of the lesion site. We're examining long-term safety and efficacy in monkeys, and we are preparing human GMP grade cells to bring into a clinical trial. And I'd like to give credit to Hiromi Kumamaru, who has returned now to Japan, who spent several years working very hard and very expertly to develop our lead human clinical cell line. This was published in Nature Methods. I think that we are running short on time, so I'm actually going to skip the next section, which was going to tell you a brief story about the transcriptomics of these grafts and how they reveal something very unique to us. I will just refer you to a paper we published in Nature last year that described the fact that these injured adult neurons that regenerate after spinal cord injury transcriptionally regress to an embryonic state. It's remarkable, again, that if you injured an adult neuron in the cortex, it transcriptionally regresses to an embryonic state, and while that state lasts, which is only a few weeks, it can regenerate. So if you're interested in that story, I refer you to Nature. So finally, I just want to mention one last thing before I wrap up, and that is the potential use of bioengineered scaffolds combined with stem cells, given that we have several bioengineers on this call. So this work was led by Kobe Kofler in our group, and we collaborated with Xiaochen Chen in the UCSD bioengineering department, together with Wei Zhu, his postdoc. And so again, I'm going to skip ahead and just mention that. So Xiaochen developed these methods of continuous projection microscale printing. So this results in layerless printing, bioprinting of three-dimensional structures that is rapid. It prints a spinal cord scaffold in 1.6 seconds. It is layerless, so it has intrinsic strength of mechanical properties and doesn't tend to come apart at individual layers. And we can 3D print these scaffolds to contain our neural stem cells to the shape of an individual spinal cord injury. So let me just show you. We designed scaffolds to mimic the bioarchitecture of the spinal cord. And then we implanted these scaffolds into rats and now monkeys with spinal cord injuries, filling these individual channels with neural stem cells. This is what the 3D printed scaffold looks like. It can be printed to fit to the dimensions of a complex human contusive injury, as you see here. And we can implant these in rats and they readily integrate. Here's the implanted scaffold. The stem cells survive when implanted into the walls of the scaffold at the time of injury. It's a very nice picture showing the green stem cells surviving within the wall, within the channels of these scaffolds. And here's why we're doing this. If we just have a stem cell graft of cells with no surrounding architecture, then the axons shown here in red that grow into the scaffold are randomly oriented. They're in all kinds of directions. But if we put them in the environment of a scaffold with linear channels, then the organization of axons growing into and out of the scaffolds is linearly organized. So this enhances the fidelity of the architecture of the stem cells and the host axons growing into them. And we are implanting these now in these contusive spinal cord injury sites and examining whether these might be an improvement in the technology that improves these outcomes. So here's a rhesus monkey spinal cord. Here's the scaffold implant on the right side, filled with neural stem cells. So we are continuing to evaluate this technology and will advance it to the clinic as findings warrant. So we expect to, this is my last slide, to begin human clinical trials if all goes well with our GMP cell lines on a timeframe of 2 to 3 years without the scaffold. The scaffold technology is following on that and perhaps could reach human utilization in about 5 years. We're also working with our colleagues in Switzerland, Grégoire Courtine, to combine neural stem cells with epidural stimulation of the spinal cord in monkeys and then in humans. We are extending our work to chronic injury to see if this will be effective. And finally, we are very interested in working with induced pluripotent stem cells. And we will establish a collaboration with Dr. Okano to push this work forward. So that's the end of my talk. Thank you for your attention.
Thank you, Mark, for a wonderful talk. The question and answers is open, and I can read your questions for Dr. Sosinski if you post them. While we're waiting, I wanted to ask Mark what you thought was the primary sort of limitation on the functional recovery. Is it the number of neurons? Is it the type of synapses? Is it the microtopography of the substrate? What do you think is the, what do you need to go after to advance that degree of functional improvement further?
Yeah, that's a great question, Andrew. And, your guess is as good as mine. We have about overall 30 to 40% recovery. And as I showed, we have about 40% of the original axon population reconstituted by the stem cells. So it could just be those numbers. It could also be that some axons find the wrong target, or that a synapse that is formed is not sufficiently strengthened to be maintained. It could be that if we combine physical rehabilitation with the graft, that we'll see a with more tuning of the circuitry and an improved functional outcome. And indeed, we have preliminary data unpublished that supports that idea. It could be that the grafts don't have sufficient intrinsic excitability, and that if we increase their excitability, they may function better. Hence the attempt to use epidural stimulation. So many possibilities that we're just beginning to work through.
I mean, your ability to sort of replicate the kind of anatomy of the spinal cord with 3D bioprinting to build substrates that act as sort of guidance for neuronal outgrowths. I think it seems particularly exciting and be interesting to see what the sort of functional advantages of that are.
Yeah, we agree completely. That work is being supported by a DARPA grant right now.
Yeah, actually, so when you transplant, so GFT labels the stem cells, we notice that's a robust axonal regenerations to compare the classical transaction model here. So, but there is some inhibitory substances, myelin or NOGO or gliascar. How do they overcome these inhibitory cues? Could you make explanation or interpretation?
Yeah, actually we can. So Dr. Okano, we published a paper in Science Translational Medicine in 2019 on just that topic. And we found that these developing neurons, which naturally do not see myelin, right, during neural development, because oligodendrocytes haven't matured yet, are not inhibited by the oligodendrocytes. Not just that, they are stimulated to grow on the oligodendrocytes. And we did RNA-seq and proteomics on the graft and on the host spinal cord, and we identified one receptor on myelin, it's called NEGR1, that mediates some of the attraction of these outgrowing stem cell axons onto the white matter. And if we knock out the NEGR1, there's a significant reduction in growth. So the molecular mechanism that in part accounts for this growth on the myelin is NEGR1.
We have a couple of questions.
Your question.
Okay.
Yeah.
Oh,
we have a couple of questions in the question and answer. Sorry, Okano.
Yeah. Please go ahead. So could you introduce Q&A?
Yeah. So Dr. Lee is asking how sort of mechanical electrical stimulation of the graft affects regeneration. So based on clinical experience.
Well, we don't know yet because there hasn't been stimulation of the graft yet, either clinically or preclinically. So we are just beginning those studies now in rats before we take them to monkeys, but we don't have results yet. So these are ongoing and future directions. You know, in theory, what stimulation might do is improve the excitability of the neurons within the graft so that it lowers their threshold to fire an action potential. And that could lead to the stabilization of synapses. That action potential driven activity of the graft neurons could also drive more axon growth out from the lesion site and into the host beyond. And once again, if it drives activities in those graft neurons, then it may stabilize those distant synapses too.
Yeah, this is a very good point. Actually, we have some similar investigation in my lab. We are submitting soon. Anyway, okay. Is there any questions?
There is one more question from Dr. Kuoka. I think you might have answered already, which is how the bioprinted scaffolds, how much are they improving the quality of regeneration?
Yeah, I think I showed that in the talk. They linearize the growth, which is incredibly important because, you know, we want to match the injured system above the spinal cord with the system below the spinal cord. And if the axon enters the graft and meanders around it, it may exit in the wrong place, or the splice may exit. So now if we align it, there's no place to go, but in the proper tract distally. You know, that's the hypothetical model. And you know, we're still investigating to what extent that might improve the function.
Well, thank you again, Mark, for really a fabulous and really exciting talk. And